start us off.
In the name of the Father, and the Son, and the Holy Spirit. God, you are the creator and the sanctifying spirit. Help us to carry out in our lives the love and example of Jesus by striving for peace within ourselves and seeking to be peacemakers in our daily lives, by accepting suffering in the struggle for justice rather than inflicting it, by refusing to retaliate in the face of provocation and violence, by persevering in nonviolence of tongue and heart, by living consciously and simply so that I do not harm creation or deprive others of the means of life, by actively resisting evil and working non-violently to abolish hatred and causes of hatred from our hearts and from the faces of the earth. God, we trust your sustaining love and believe that just as you gave us the grace and desire to offer this, so too will you bestow abundant grace to fulfill it. We pray for peace in your name. Amen.
Every community owes its existence to vitality to generations from around the world who contributed to making the history that led to this moment. We recognize that all of us at Bellarmine learn, live, and work on or near the unceded territory of the Coast Salish people. In particular, our campus is situated on the traditional lands of the Puyallup tribe. As we gather, we also want to acknowledge the lands of the Muckleshoot, Nisqually, Squamish, Snohomish, Shemamish, and Duwamish, and all traditional lands on which we live and work. We want to acknowledge the histories of dispossession and forced removal and reflect on this history in ways that honor the memory of those dispossessed and keep us committed to the cause of social justice for Indigenous populations. And here's Mr. DeGroat again.
Well, again, thanks for being here tonight. If you took a look at the program, you'll see a variety of projects, as usual. Our program will be, we're going to do 4 presentations, take a little break, and then finish up with 3 more. I'm going to ask the students to introduce the next presentation, so I won't be up here too much, but I'll finish this off at the end. There will be time for questions. So I've already told the students this, ask them hard questions, okay? Really test what they learned. So without further ado, Katie Vaughn is going to come up and talk to us about what's under the water in her neighborhood.
Hi, my name is Katie Vaughn, and my presentation is on the comparison of 2 artificial reefs' ability to recruit organisms in Carr Inlet. So, for the background, artificial reefs are a vital part of the ocean's ecosystem, and since coral reefs are dying, artificial reefs are becoming more important to restore marine habitats and make sure that all marine organisms have a place to live. In the Puget Sound, however, in the 1970s, artificial reefs were made by the Washington Department of Fish and Wildlife, and they had dumped over 500,000 tires into the water. And at the time, they thought this was a really good idea to help the marine ecosystems, but Now they've actually realized that the tires are leaking 6 ppd quinone into the water, which is a really dangerous toxic chemical, and it's hurting many of the animals, but mainly the coho salmon, which is a really big problem since they're really good to the Puget Sound. And so for my project, I decided to find a cheaper way to build an eco-friendly artificial reef to help implant in the future as a way to replace the toxic tires. So my problem was which artificial reef, the PVC pipe or the concrete drainage blocks, will attract more marine organisms. And my objective of the study was to measure the abundance and diversity of organisms attracted by the artificial reefs with a possibility of replacing toxic materials. So I hypothesized that the concrete artificial reef would attract more organisms and be better to implant in the future based on future research. So I had 2 mentors. My first mentor was Stina Troyer, who works at Harbor Wild Watch, and she helped me identify all of the animals and put them into spreadsheets. And my second one was Dr. Greg Davis, who did all the diving for me and collected all the video footage for me. And my role as the student researcher was to design and build the reefs, as well as pick the placement in the water. And I also had to analyze all the video footage, and I put them into spreadsheets to use for my results. So I used 18 by 18 concrete drainage blocks, and they originally didn't have holes cut out of them. So we had to cut holes out of them so that it'd be easier for organisms to swim through. And I transported it by paddleboard out to the water, and then I dumped it into the water, so my diver later then rearranged it underwater into a pyramid shape. And for the PVC pipes, we just cut the PVC pipes, and then we drilled them together to make a pyramid-like shape that my diver took out to the water also and placed almost as close to the concrete artificial reef, but farther away so that it wouldn't be right next to each other. And some of my results from December, March, and May, the concrete artificial reef is on the top, and the PVC pipe one are on the bottom. And December was the installation date, so there's not really anything on in December. And then July, August, and October were the other 3 months that I had data for. And you may notice there's no August for the PVC pipes, and that's because we actually lost it. We couldn't find it. So because of this, we had to make a trail of golf balls attached to a rope so that it would sit at the bottom of the ocean floor so that we didn't lose it again. So my first 2 graphs are about the biodiversity of the artificial reefs because they were in the same ecosystem and they were relatively close to each other, making them easy to compare. And as you can see, the PVC pipes had more of the same animals coming at the same times, whereas the concrete artificial reef, if you see the dark blue, is molted starfish. And there was more of those on the concrete artificial reef coming back daily than there was in the PVC pipes, which just came back in a couple months and then just disappeared and didn't come back again. And then similar to the last graphs, these are my seaweed covered percentages. And in my results, in my pictures, you could see how much of them was covered by seaweed. But as you can tell by the graphs, the seaweed is on seasonal changes. So in December, the installation date, there's no seaweed because it was just input into the water. But then as you get into the summer months, like July and May, it increases rapidly. And then there's no August again because we couldn't find it. But then once it goes down to October, it's decreasing because the seaweed goes away in the winter months. So I used the Shannon Index, which measures the biodiversity of the recorded area. And as you can see, both of them have the same richness in number, but the higher the number for the Shannon Index on the very top part, the less diverse it is, meaning that the PVC pipes was more diverse. It had more different animals coming time after time, but the concrete drainage blocks, artificial reef, had the same animals coming back time and time again. So I was limited to concrete and PVC pipes because those were the only things I had budgeted for, and in my research, they were the best things to use for making an artificial reef. And if I did this again, I would do access to other locations because I only had location to Carr Inlet. I didn't have time to get a permit to go to another place. And then for access to diving, we didn't have a set diving schedule. So some days it was 2 months in a row or it was every other month. So we weren't always exact because the weather also, we had to cooperate with if it was going to be stormy out or not. And I would definitely mark the artificial reefs in the beginning next time so that you don't lose it and lose a whole month's data. And then with the seasonal changes, the water wasn't always the clearest. So some of my video footage was murkier than others, leading to some estimated calculations. And this picture shows one of the artificial reefs, and you can't really see it at all because the water was so murky. And for areas of future research, in the future, I would use other materials like either steel or some other type of rocks. And I would try more locations to see if it's only good in inlets or if it's good in the open water also. And then I would also put an artificial reef close to one of the toxic tire reefs because you would want to see the comparison between a non-toxic reef to a toxic reef to see how the non-toxic reef is a way better thing to implant in the future. And then I would frequently analyze the reefs every other month or every month to see if there's any big or small changes to them. And then also for the future, I would consider continuing this project to see another year's worth of it, to see if it's the same species coming back and the same seaweed percent total coverage, just to see how the cycle works. And my picture is the concrete artificial reef in November, but it wasn't in my data because I had already collected all of my data by the time I had the video. And I'd like to thank the Marine Chem class of 2024 for a great 4 years together and a great time in Hawaii. And then I'd like to thank Stina Troyer for helping me identify all of the animals and analyze the videos. And then I'd like to thank Dr. Gray Davis for diving and collecting all the video footage for me. And then obviously my family for helping me through the entire process and just being there to support me. And then Mr. DeGroot and Mr. Nielsen for helping mentor us through our 4 years and with all the papers and letting us have a great time in Hawaii. Here are my references. Any questions? Yeah. Can you say that a bit louder? So I had done research on this, and it said that those were the best, or the question is, how do I know the PVC pipes and the concrete pipes, or the concrete drainage blocks aren't toxic? It's because I had done future research, and there's other research done, and it's called the Reef Ball program, and it's the same concrete system, except that it's a ball of concrete with holes in them. So I already knew that the concrete was good, and then I just researched if PVC pipes were good, and they don't release any toxins, and they were cheaper to buy than like buying steel to make it. So that's how I decided to use those two. Yeah. So they were, I'm just going to assume that they were at the same depth. It was at 2 buoys, which are at the same depth. So I'm just going to assume that they were because they were relatively close to each other. I don't really think it would have made that much of a difference if one was deeper than if not, because at some point, if it was closer inland, it probably wouldn't have gotten as many, just because when you go to the beach and you see the tide and it's way out, you don't see as many animals. So if it was closer in, I think that would have made a bigger difference. But since it was already really far out and it's never going to be beached on a really low tide, so I don't really think it made a difference. So next up we have Maya Deaton and Talia Patel on crab larvae.
My name is Talia Patel, and this is Maya Deaton, and our project is on the development of an identification key for improving accuracy of larval crab population counts in the Puget Sound using DNA barcoding.
Native crabs are crucial to our ecosystem. With 4 life cycles of zoea, megalope, instar, and adult, they are crucial as they could potentially initiate the collapse of the Puget Sound ecosystem if populations were to dwindle. Dungeness crabs revealed that they are vulnerable to low pH, low oxygen, and high temperatures. This means that monitoring crabs is essential to preserving the Puget Sound ecosystem. As an $86 billion industry in Washington state in 2022, the commercial crab fishing industry is dependent on population counts. As previously mentioned, native crabs have 4 life cycles as shown here. They start out as zoea here on the right, then develop into megalope here at the bottom, and then develop into instar up at the left, and then finally reach adult up at the top.
Past research done in Oregon shows that larval crab counts predict future adult populations. Using this information, the Pacific Northwest Crab Research Group, or PCRG, created a method to collect larval crabs utilizing light traps dispersed throughout the sound. PCRG is a coalition of native tribes and scientists working to preserve crabs in this region. But volunteers check the traps. Crab species are difficult to identify at the larval stage by volunteers, and therefore PCRG cannot predict species-specific future crab trends. Research conducted in the Red Sea used DNA barcoding with morphological characteristics to create a field guide for crabs in that region.
The problem that we encountered is larval crab populations cannot be accurately recorded in the Puget Sound due to a lack of field guides available, not allowing the species to be identified in the field. In efforts to correct this issue, we work to collect and identify crabs so that we can create a field guide containing their distinguished features so population counts may be improved. We believe that in creating this field guide, it will allow people without a technical background to be able to identify crabs to provide accurate species data to PCRG.
So our mentor was Jessica, and she was a student at University of Washington, Tacoma. And she showed us how to do morphological identification and collection and helped us create and test the field guide with volunteers.
On the left of the slide is a map of all of the PCRG trap location sites. The mass center is located at Redondo Beach in between the East Passage and Commencement Bay. We selected this site as it had former experience deploying light traps and a provided lab where morphological identification and DNA analysis preparation occurred. We deployed our light trap off the mass center pier using a pulley system. The pulley system ensured that the trap would stay in the water as the tides changed. We used the trap for collection from late spring to the end of summer.
So this trap was built following PCRG format, as you can see on the right. So crabs exhibit a trait called positive phototaxis, which means they're attracted to light. And so their eye spots at the larval stage cannot form images, but enable them to sense light. So a timer was used from sunset to sunrise every night in the summer to collect crabs overnight. Our image is not loading. Okay. Okay, moving on. So on the left, we have an example of a zoea and how we would take note of it in the field. So once we would get the trap, pull it up in the morning, we would observe the crabs under a microscope and then take note of them. So on the left, the zoea, you can see that we would take note of the abdomen, carapace, and dorsal spine, as you can see. And on the right, we would look at a few different characteristics for megalopae since they have different anatomy. So we would pay attention to the antenna, and then a separate carapace color, and then their legs as well. And then once we would take note of those crabs and take pictures of them, we would put the samples of the crabs into ethanol and preserve them to send them to Seattle University. So at Seattle University, the COI gene was extracted and inputted to the Barcode of Life data system. So this gene is frequently used as the marker of choice because no other genetic region can be found in taxonomically verified databases with sequences covering so many species. So once we would have the species returned to us, we would match the note with the species that we received.
We modeled our field guide based on the PCRG format for volunteers to potentially use in the field. In total, our field guide contained 8 species of crabs. Each slide is dedicated to a specific crab species. It contains the species name and common characteristics that each species has. We used selected photos that showcase good angles, clarity, and orientation of the crab. In testing the field guide, we recruited 13 volunteers with no crab identification experience. We created a test to determine the field guide's accuracy. Each question had a photo of a larval crab and asked the participant to identify the larval species and the larval stage using the field guide. Each participant worked alone after receiving a hard copy of the field guide. Prior to the test, they were provided with a copy of zoea and megalopae anatomy to understand the anatomy used in the field guide. After completing the test, they gave us feedback. Here is a page taken from the field guide that we created. A key thing to notice is that for both zoea and megalopae, key characteristics were noted. The pictures that we selected showcase these characteristics clearly. After our field guide experiment, we calculated the accuracy for correct answers for the species, as well as the correct answers for the larval stage. The larval stage had 100% accuracy. The larval species received an 80.625% accuracy overall. However, accuracy varied for different species. A positive thing to note was that the crabs that had more defined characteristics and are easier to identify received high accuracy percentages, versus the ones harder to identify, which received lower accuracy percentages. Looking at the results table, there are the 8 different species listed in the top row. In the second row, the percent accuracy is recorded. The 2 crab species that the volunteers struggled with the most were the hairy red rock and the yellow shore crab. In the third row, we indicated for the lower accuracy species which answers were given instead of the correct answers.
Discussion. So in reviewing our field guide, we shared 3 species with PCRG's current megalopae field guide. So we had in common the pea crab, pygmy rock crab, and the graceful decorator crab. So something to note that's different about our field guide is that we had obviously different species, and as well as we included the zoea stage. So our hypotheses were mostly supported. We were able to distinguish unique characteristics in larval crabs, as well as being able to test it on volunteers with an accuracy rate of over 80%. So this accuracy rate suggests that PCRG should consider using volunteers for species-specific crab larvae data in the future. So some of the limitations we ran into is that we only collected crabs from one survey site. So the Puget Sound has over 4,000 kilometers of shoreline with changing depths, acidity, and temperature, which means different crabs will be in different parts of the sound. So increasing survey sites that use DNA barcoding would lend to a broader range of species that we could add to the field guide. Additionally, we only were able to test our field guide to volunteers using photos because it was not in the light trap season. So using this field guide during real field work through a microscope where volunteers can look at would be more helpful and would give us more information to how helpful the field guide is in a real field situation. And our findings, paired with possible research, offers promising hope for the future of Pacific Northwest crabs. Thank you.
Thank you to our mentors, Jessica and Dr. Becker, the Becker Lab Group, Ariel from the Mass Center, both of our families, Mr. DeGroat and Mr. Nielsen, the RRC, which was the best van in Hawaii, room 626, and the Marine Chemistry class of 2024. And here are we're excited. Any questions?
Yes, right here in the front. That's a great question. So the question was, how many samples did we get over the course of pulling the trap? So every day when we pulled the trap, which is about 3 times a week was the goal, we would still get like a sample every morning of like a new haul, obviously. But the crabs that we actually sent to Seattle University, we had I think we had around like 30 to 40 samples that we sent out. And we got 8 species from that to include on this field guide. Any other questions? Oh, yes. So the trap was collected about 3 times a week. So they were about 3 times a week in the morning. I think it was Saturdays, Mondays, and Wednesdays, and I think maybe Fridays. It kind of changed from there. But yeah, we would only check the trap 3 times a week. And Maya and I weren't able to be there for every trap. We tried as much as possible, but other volunteers would then take over, but they would not do the morphological work like we did. They would just check the trap. Yes. So the question was, did we get the field guide published and do we have a copy? So currently, no, the field guide is not published. I know my mentor, Jessica, and the rest of our lab group is working with other people to get that published. And currently, right now, we worked with PCRG. That's who our lab is partnered with. So currently, we are not able to share the field guide. Yes, Jessica. Yeah. So the question was, how would this research help with future problems with the European green crab, which is currently an invasive species in Washington right now? So the European green crab, it essentially can thrive in warmer water, and therefore it can be dangerous and kill off native crab populations like the Dungeness by taking over its habitat. So for volunteers being able to identify the correct crab is really important. So maybe if they don't see like a certain crab, it might be a red flag, and they could like talk to more like scientists about it and say, hey, like what about this crab? And this would help for scientists in Washington to know where the European green crab is at one point. Yes. No, you're good. Yeah. So we did not conduct any of the actual DNA barcoding. That was all Seattle University. So from there, we wouldn't know the exact species that we put in that vial. We would just take notes of it. So we would take as many specific characteristics as we could of a specific crab, put it in a vial, send it to Seattle University. So there was another person there who helped us out, and they would extract the gene, and from there they would tell us the exact species that was in that vial. So once we would know the exact species, we could match it to the characteristics that we had previously recorded at the Mass Center. Yeah. Any other questions? Perfect. Thank you. Okay, up next is a great friend of mine, Amaris. She's amazing. She always walks in with like a huge smile on her face, and I love her energy. So you guys are lucky that she's presenting for you. Go Amaris.
Hello everyone, my name is Amaris Alstead, and tonight I will be presenting on determining the presence of Phytophthora species in soils of diseased alders at Kopachuk State Park and Point Defiance Park. Phytophthora species are microorganisms that cause diseases in various plant groups. These species have caused devastating economic losses on crops worldwide, as well as environmental damage in natural ecosystems. Phytophthora was the primary cause of the potato blight that caused the Great Famine of Ireland in the mid-1800s, which killed over 1 million people and forced 2 million to leave Ireland. Phytophthora remains the most destructive pathogen to Solanaceae crops, such as tomato and potato plants. Phytophthora is especially widespread in nurseries. A particularly dangerous and quick-spreading species that infects alder trees, also known as Phytophthora alnii, is a single pathogen made up of diverse genotypes, having been accidentally created in the nursery in Europe. Although this species has not been found in the Pacific Northwest, it is important to continue to regulate and test different alder populations to ensure overall health and to hopefully reduce outbreaks by early treatments. Symptoms of Phytophthora can include root rot, leaf blights, stem dieback, and bleeding cankers. Species kill the plant's tissues by infecting the roots and making their way upward, with the most noticeable effect on the upper canopy due to lack of nutrients. Most commonly, Phytophthora spores move between plants through water systems, which causes crops and trees located near rivers and streams to be most affected. The Phytophthora then forms hyphae and contaminates the tree's water supply. As it grows, it fully infects the plants, producing one or more spores to begin its life cycle again. In 2022, Gilhan Baker reviewed large old trees with a focus on ecosystem preservation, climate issues, and therapeutic potential, and found that they had a positive impact on the overall health of the forest through carbon dioxide storage and nitrogen fixation. Specifically, alder trees are critical to the health of Western Washington's ecosystems as they provide shelter for various animal species, are erosion control for riverbeds, and are vital for nitrogen fixation. Several studies by Western Washington University show that alder trees have become a short-lived species and are aging out of several forests for unexplained reasons, potentially causing a negative chain reaction among other plants and animals that depend on the alder tree for shelter and food. Research done by Washington State University has determined that Phytophthora has spread through water, soil, and vegetation in Pierce County, Washington, after first being detected in a nursery in 2003. Dillo and Iea, former Bellarmine students, completed research in 2021, which proved that Phytophthora was present in dying alder trees at Titlow Beach Park. Phytophthora pseudosyringae, Sikorensky, and Camivora were all found at the sites they tested. More research must be conducted to determine if Phytophthora is a widespread cause of alder tree death here in Western Washington. My first question for Phase 1 at Kopachuk State Park was how widespread is Phytophthora species outside of nurseries in Western Washington? I hypothesized that Phytophthora species will be present in alders showing at least 25% of branch dieback. My second question for Phase 1 was what symptoms of alder trees and other characteristics of this study indicate Phytophthora contamination. I predicted that sites with access to running water will have a higher percentage of alder trees infected with Phytophthora. My questions and hypotheses were similar for Phase 2 of my project at Point Defiance Park. The methods for Phase 1 and Phase 2 are identical except for the process of choosing the location for each phase. For Phase 1, I chose Kopachuk State Park, where there's a great presence of alder trees with nearby running water which could facilitate Phytophthora growth and reproduction. On the other hand, Point Defiance Park was chosen as the location for the second phase, due to the few rivers connecting alder trees, which may suggest a lower presence of Phytophthora. 4 trees were chosen from 4 sites at each location. Soil samples were taken from around the base of the tree. Each sample was labeled in the format of Site 1, Tree 1. This labeling system was used throughout collecting, fading, isolating, and plating. The trowel was wiped off to prevent contamination between sites and trees. Roughly an inch of soil was placed in a clear labeled plastic cup. 2 rhododendron leaves were placed in each cup, and it was filled with deionized water. After 72 hours, the rhododendron leaves were then placed on a wet paper towel and in a plastic bag to continue the growth process. Rhododendron leaves were specifically used as bait for Phytophthora because they are a very susceptible host and encourage Phytophthora growth. After 5 days, the Phytophthora leaves grew colonies of, or the rhododendron leaves, grew colonies of Phytophthora. 5 areas of those leaves were hole punched and put into a properly labeled VA agar petri dish. Within 2 to 4 days of the plating process, the edges of the bacteria growths were isolated and put into a VA-only agar plate. Once the isolates were grown, they were viewed under the microscope for identification and set to be DNA sequenced to determine which species was each isolate. The tree health column on the second column from the left side provides an indication of the health of the trees sampled from each site. For example, for Site 1, 2 out of the 4 trees were sampled had a 0 to 50% canopy dieback, and the other 2 trees had a 50 to 100% canopy dieback. Phytophthora chlamydospora was found in 3 isolates from Site 1. Pseudosyringae was found both in one tree sample from Site 2 and in 2 tree samples from Site 4. Camivora was found in 1 tree sample at Site 3. During the second phase of research, no Phytophthora species were found at Point Defiance Park. Pythium, a pathogen very similar to Phytophthora yet less destructive, was found at each site. These 3 pictures were taken of isolate 1-1-1, 3-3-1, and 2-2-1 respectively. There are slight differences in the structures of these 3 species of Phytophthora, which helped us identify which species was found at each site and tree. Chlamydospora has right-angled branching with constricted bases, where Camivora's hyphae are flat and irregular. Pseudosyringae have more knobby hyphae than the other species. Determining which Phytophthora species is present in Washington state soils is crucial in order to ensure the overall health of forests and to reduce outbreaks by early identification and control, specifically of Phytophthora alni. My hypothesis for my first research question was not supported by my data. The alder trees that had Phytophthora had a range from 0 to 100% canopy dieback instead of the 0 to 25% that I originally predicted. Phytophthora camivora was found in a tree that showed signs of dying, suggesting that this species is overall more harmful to alder trees. Out of the 3 species found, Camivora is the only known pathogenic species, which supports the fact that it was found in a dying tree. Pseudosyringae was most commonly found in the soil of healthy trees compared to unhealthy trees. This confirms previous research done by Dillo, Iea, and Su that Pseudosyringae does not cause severe harm to alder trees. Additionally, this suggests that Pseudosyringae is native to the Pacific Northwest because it does not cause serious disease. This species could potentially cause harm if introduced to the southern hemisphere where plants have not developed immunity. Since chlamydospora was found at 3 out of the 4 samples in one site, this species has most likely infected this site completely. It is a known weak species, which may prove why this species was found in trees despite their overall health and canopy dieback. Phase 2 samples were collected before any significant rainfall had occurred for the season, which may be the reason why no Phytophthora was found at Point Defiance Park. Additionally, although efforts were made to ensure limited cross-contamination between sites, it is possible that soil from one site or tree was transferred to another on the trowel, which may have skewed the data. Although the absence of Phytophthora in Phase 2 was initially surprising, after comparing Point Defiance Park and Kopachuk State Park and noticing the extreme differences in the 2 locations, The results from phase 2 make sense and even confirm popular scientific studies about how Phytophthora moves and reproduces. For example, Point Defiance Park had overall healthier sites and alder trees than Kopachuk State Park, which explains the lack of Phytophthora species found. Additionally, Point Defiance Park is a more mature forest and therefore has fewer alder trees and more distance between them. Point Defiance Park also had less running water near the alder trees than Kopachuk State Park. This is a very important difference because Phytophora's main travel method is through flowing water. Furthermore, soil samples from phase 2 were taken earlier in the season than soil samples for phase 1. Due to the dry 2023 summer and no major rainfall prior to the sampling for phase 2, Phytophthora spores had less time and ability to spread through the sites. This comparison between the parks does support my hypothesis for the second research question, that sites with running water are more likely to be infected by Phytophthora. Although unlikely, it is possible that Phytophthora from the soil samples in phase 2 did not properly infect the Phytophthora, the rhododendron leaves, and therefore was not present in our analysis. Hundreds of species of Phytophthora have been identified, yet hundreds more have yet to be discovered. So continued research on species identification will prove valuable when surveying and protecting against future Phytophthora outbreaks, specifically Phytophthora alni. The difference between the results at the park can be attributed to several factors, such as the levels of flowing water and the maturity of the surrounding forests, which all, if future research is conducted, may provide important insight about the growth patterns of specific species of Phytophthora. Furthermore, the same sites at Kopachuk State Park and Point Defiance Park can be sampled seasonally or yearly to determine when and how Phytophthora moves and reproduces. Treatments such as chemical controls and pesticides must also be tested to determine their efficacy both in nurseries and in more natural locations. Throughout both phases of my research, there is an indication that Phytophthora species, specifically Phytophthora camivora, is a cause of disease and infection, which can be corroborated through previous research. Furthermore, Phytophthora pseudosyringae and Chlamydospora are milder species of Phytophthora due to their presence in healthy trees as well as unhealthy ones. Phytophthora spores were found mostly in healthy trees, which signifies that native Phytophthora species may not cause harm to native plant species in the Pacific Northwest. The lack of Phytophthora at Point Defiance Park confirms previous research on how Phytophthora moves and reproduces, specifically through water systems. Here are my references. I would like to thank Dr. Marianne Elliott for her invaluable time and guidance through the completion of this research project. I would also like to thank my parents for their support, Mr. DeGroat and Mr. Nielsen for their assistance, and of course, my Marine Chem class of 2024 for their encouragement. What questions do you have? The question was about how you treat Phytophthora once it is found in alder trees. So many methods for treating it is very complicated and expensive, such as steaming and any chemical controls or pesticides, and those are usually only specifically used when you have important alder trees that you want. So other methods that you can maybe do in a park that has a large amount of Phytophthora that has been proven to cause the dying trees is you can improve the water drainage systems, and that would probably most effectively help stop the Phytophthora from moving through the water from tree to tree. The question was, how do you ensure that the rhododendron leaves were not a cross-contamination? I'm guessing for the Phytophthora that would grow on those leaves. So the rhododendron leaves those bushes were all healthy where I took those samples from, and the soil was also healthy surrounding those. And specifically for what I was testing, those Phytophthora species are not infected inside the rhododendron bushes themselves, and the leaves are only susceptible to that growth in that process. Any other questions? Can you say that one more time? Can you repeat your question? So the question was, how do I know that the rhododendron bushes themselves were healthy? I did not test the rhododendron bushes, but they were part of a Washington State University lab and nursery, so that they were known and they were reliable sources of where I could get those rhododendron leaves for my project. Any other questions? Thank you. Our next presenter is one of the sweetest and smartest people I know, Maya Cratley.
Hello, my name is Maya Cratley, and my project is a comparison of sediment microplastic concentrations in marine protected areas of the Puget Sound. Since the 1970s, researchers have been alarmed by the abundance of microplastics in the ocean. A 2015 study estimated that there are around 14 million tons of microplastic on the ocean floor today. So naturally, I was curious how this pollution problem was impacting our Puget Sound. As defined by Missouri et al. 2015, microplastics are plastic particles ranging from 0.33 millimeters to 5 millimeters. They are classified as depicted in the image above as fragments, pellets, films, foams, and fibers. Primary microplastics are intentionally produced in this size range, as seen often in microbeads that are produced for cosmetics, and these are mostly classified as pellets. Secondary microplastics, on the other hand, break down from larger plastic pollutants and are the majority of the microplastics that I'll be studying here today. Due to their prevalence and minuscule size, microplastics are commonly ingested by marine organisms and passed up the food chain via biomagnification. Iwalai et al. 2024 observed that following exposure to microplastics at environmentally relevant concentrations, amphipods demonstrated prey intake reduction that could affect reproductive outcome and growth. Additionally, fecal pellet density increased following microplastic ingestion, diminishing fecal pellet nutrient exchange in surface waters. These changes in ingestion rates and prey consumption were noted to be more significantly affected by microfiber concentration than exposure time, and concentration is the metric that we'll be using in this study. Microplastic concentration has additionally been shown to correlate with other variables in the Puget Sound. Serdan et al. Observed that microplastic concentration is positively correlated with population density. Mahoney indicated that it is more closely related to residence time than population density. And then Hall 2018 additionally found that it varies spatially along the Puyallup River, and that concentration overall increased from 2017 to 2018, as shown in the image above. Additionally, it is theorized that Puget Sound has saltwater upwelling and tidal exchanges that produce a low flush rate of water, so that low flush rate might cause pollutants that are trapped within the shallow sills to maintain a higher concentration than in the open ocean. In a measure to conserve marine ecosystems, the 11th HE target of COP10 stipulates that at least 10% of coastal and marine environments be conserved through marine protected areas, or MPA systems. This need for conservation has increased since 2014 due to concerns over the loss of biodiversity. In 2021, Kutrala Munyasamy found that marine protected areas cannot protect from microplastic concentration. Out of the 36 different MPAs they studied, microplastics had permeated 29 of them, and 50% of the studied biota had actually ingested microplastics. In 2022, Nunes et al. Took this a step further and found that globally, microplastic concentration was actually greater in marine protected areas than it was in unprotected areas. And MPAs had an average microplastic concentration in the water column of 12,429 items per square kilometer. Washington has a system of 127 MPAs that are managed by 11 different governing agencies and spanning 644,000 acres. They're classified as conservation areas, as shown by the red image above, which means that the extraction of certain marine resources is prohibited. And in this study, we studied the conservation areas Brackett's Landing and Richmond Saltwater Park. We also studied a marine preserve indicated by the blue. And a marine preserve, the extraction of all marine resources is prohibited. And so we studied Titlow Marine Preserve. This study aims to fill in a research gap and a lack of data on the impact of microplastic concentration, or impact of marine protected area status on microplastic concentration in the Puget Sound, in hopes of better informing Washington state regulations regarding marine protected areas. So my first problem statement was that it is unknown what impact marine protected area status has on microplastic concentrations in the Puget Sound. And my objective was to determine that thing from sediment data in local areas, protected areas, and unprotected areas. My hypothesis was that marine protected area status would have little to no consequence on the microplastic concentration because of the findings in Nunes et al. And the data that suggested the Puget Sound has a low flush rate and a lot of tidal exchange. My second problem statement was that microplastics come from a variety of primary and secondary sources, and since this is a relatively new field, there's little data in the Puget Sound on the different microplastic types, and so I hope to categorize them by type and then enhance the understanding of local sources of microplastics. My hypothesis was that fibers would be overwhelmingly the most concentrated microplastic type because of past University of Washington-Tacoma data sets. So my first form of methods was site selection, and so for the site selection, we referenced Christie et al. 2014, which is a NOAA study that actually looked at the degree of enforcement of different marine protected areas in the Puget Sound. So in their study, they did a bunch of different surveys of local populations to determine what the perception of marine protected area efficacy was, and in this, they used the idea of a reference site, which is essentially a nearby location to a marine protected area that has similar usage, but has no regulations like a marine preserve has. So you can see here on the left, we have the marine preserves that were studied and the reference sites that were selected. And you can also see that in the stations map above. Samples were collected via a Van Veen grab at depths ranging from 10 to 15 feet, 20 to 30 feet, and 30 to 45 feet for variation. We also wanted to make sure that all of the depths were around the same for all of the different locations so that there was consistency in that. You can see here in the diagram on the left, the Van Veen was lowered from a boat and then triggers once it reaches the bed sediment and then extracted sediment at a depth in the Van Veen that we've estimated to be 3 centimeters. For sample processing, we used the methods outlined in Missouri et al. 2015, which has become the NOAA standard for processing microplastics. The first step in this is disaggregation and sieving. So this is basically to disaggregate all of the sediments using potassium metatungstate and then sieve them in a range between 0.33 and 5 millimeters, which is the established parameter for microplastics. We also did a density separation using lithium metatungstate, and so all of the organic material would sink to the bottom and having microplastics rise to the top. The floating solids were then put through, and they were, and in between each of these steps, we would put them in an oven at 90 degrees Celsius overnight, and then we would oxidize the rest of the microplastics using 30% hydrogen peroxide. And then we did a second density separation step, and so essentially all the stuff on the bottom you can see is like dissolved salt, or is undissolved salt and organic material, and the stuff that is floating in the top is microplastics, and that was what we put in the sieve and analyzed under the microscope at 40 times magnification. So as you can see here, these are some examples of what a microplastic would look like under the microscope. This first one here is a clear film that we estimated to be about 1 millimeter. And so it might look really difficult here to determine what actually is considered a microplastic, because we do have some organic material that was not sieved out through the rest of the processes. However, because it went through all of those processes, the organic material is very brittle. And so it's easily torn apart by the tweezers. And so then we can theorize what are indeed the microplastics and put those in vials and weigh the vials. You can also see here a clear fiber bundle. I'll be talking about these more, but these are essentially really tangled masses of fibers that can make some data analysis difficult. This was the calculation that we used to go from microplastic counts per the wet weight in grams to microplastic counts per square meters, which is what we can use to analyze in terms of other studies, or comparison of other studies. So this is a box and whisker plot. I apologize for the white. This shows the difference between Sundrock and Lilliwaup State Park, Sundrock being our conservation area or marine protected area. As you can see here, Lilliwaup State Park had both a higher mean and a higher median microplastic concentration than the samples collected at Sundrock. You can also see a value up there at like 22,000 microplastics per square meter, and this very high variability was due to the amount of fiber bundles present in that sample. But as you can see here, overall, the reference site actually had more microplastics here, which supports the theory found in Nunez et al. Titlow also supported this hypothesis with a mean and median being greater, mean and median microplastic concentration being greater at the reference site of Chambers Bay than Titlow. You can also see here there isn't a lot of high variability going all the way up to 40,000 microplastics per square meter, and this was again due to the presence of fiber bundles. Oh, sorry, I think these got switched around. Let's come back to that one. So Brackett's Landing was actually the anomaly in this study. So in Brackett's Landing, the marine protected area actually had a significantly higher microplastic concentration than the reference site. As you can see there on the left, the blue is way higher, both the mean and the median. You can also see here on the right that the median microplastic concentration for Richmond Saltwater Park was in fact 0 microplastics per square meter, and that was the only site at which we found that, which is very promising. For the statistical analysis, we did a paired t-test, which is essentially where you take the difference in the mean microplastic concentration at the marine protected area site and then subtract it by the mean microplastic concentration at the reference site. And then we took those values of the differences and conducted a t-test at 95% confidence level. We found a p-value of 0.16, indicating that there was no statistical significance in the difference of mean microplastic concentrations in marine protected areas and the reference sites, meaning that my first hypothesis was kind of supported and that there was little to no consequence of marine protected area status on the microplastics. It was really interesting to look at this in the context of Christie et al. 2014, as Christie et al. 2014 classified Sundrock and Brackett's Landing as enforced marine protected areas, while Titlow was classified as a non-enforced marine protected area based on a scale of survey responses from community members and informants. Interestingly, Titlow Marine Preserve had the lowest microplastic concentration among the sampled marine protected areas, despite being labeled as non-enforced, whereas Brackett's Landing had a greater microplastic concentration than its reference site and was indeed labeled as enforced. Additionally, length of time under the marine protected area classification did not correlate with microplastic concentration. Sundrock and Titlow were both founded in 1994, whereas Brackett's Landing was founded back in 1970, but it still had a higher microplastic concentration. Additionally, all of these three had similar ratings of dive usage according to Christie et al. 2014, so that was not a factor. The last factor that we looked at, because past studies had looked at population density, so Mason County, which is where in Hood Canal by Sundrock and Lilliwaup State Park, has a density of about 50 to 100 people, and then Snohomish up by Brackett's Landing and Richmond Saltwater Park has a density of 100 to 200 people, and then Pierce County, where we're at with Titlow and Chambers Bay, has a density of 400 to 700 people per capita, and so but in Pierce County, we actually had the marine protected area with the lowest microplastic concentration in comparison to samples from counties that had much lower population densities, so indicating there was no trend in the study between population density and concentration. Let me go back here. Sorry, these got switched around. So my second hypothesis was about microplastic type, and I hypothesized correctly that fibers would be by far the most abundant microplastic type. Overall, our samples had 93.6% fibers, 1.5% fragments, 3.4% pellets, and 1.5% films. As you can see here, however, there was a variety in distribution in between marine protected areas and reference sites. So in the marine protected areas, about 63% of the microplastics sampled were fibers, whereas in the reference site, upwards of 96% of the microplastics sampled were fibers. And this again was due to a large amount of fiber bundles, which were estimated to have 10 fibers per bundle that were found almost exclusively in reference sites. Wait, one more. Okay. So why does this matter? So it's really interesting if you can look at it in comparison with Kunchala Munyasimi 2021, which found that in the zone 2 that was sampled, which was about all of the Americas, found sediment microplastic concentrations ranging from 178 to 3,504 items per square meter. And this compares with our estimated average microplastic concentration in marine protected areas of 977 microplastics per square meter. So we were actually fitting right in the range the Puget Sound was in terms of the greater context of marine protected areas in the Americas. It's really interesting to analyze this from the perspective of microplastics because they are a definitively man-made pollution source. So as with other pollutions like a lot of different types of runoff, you can point to different environmental or natural causes. Plastic is solely a human-made product, and so you can look at the direct impact that we are having on marine protected areas, which are areas that are designated to provide solace for vulnerable ecosystems. So then again, we have to look at the proper enforcement of marine protected areas. It's really easy to label an area as protected, but it's really important to know what it's protecting from, and the study clearly found that marine protected area status is not protecting from microplastic concentration in the Puget Sound, which raises the question of what impact microplastics are having on vulnerable ecosystems. The main limitations in the study in terms of resources was the efficacy of the Van Veen. So about every 1 in 6 times that we lowered it would be a success. And we couldn't really lower it any place that was too rocky, as the Van Veen wouldn't close all the way and grab sediment, or any place that was too heavily sloped, so that did impact where we sampled from. Another impact with resources was the QA/QC coloration. So the quality control is something that is a little box with like a glass fiber thing that we put outside of the microscope. And there, any microplastics that are falling from the natural environment in the lab will be shown, so that we can subtract that count from the total microplastic count to be sure that I'm not like contaminating the sample with my lab coat or anything. But we only used one that was white, not one that was black, so we might have missed some white fibers, and there might have been an overcounting there. In terms of methodologies, we only collected from 3 of the 127 different marine protected areas in the Puget Sound. So there was still a variety of research to be done on different areas in the Puget Sound, and also collecting at different depths. So that we collected at depths from 10 to 15 feet, 10 to anywhere to 50 feet, but we don't really know anything about sediment microplastic concentrations deeper than that. Also the different types of marine protected areas. I forgot to mention this earlier, but Titlow is classified as a marine preserve, and it also had the lowest microplastic concentration, so it'd be really interesting to look at if conservation area versus marine protected or marine preserve has an impact on microplastic concentration. This is applicable to marine protected areas solely in the Puget Sound. We didn't study any from the open ocean, but again, that would be a really interesting source of further research, is looking at the difference in marine protected area microplastic concentration in the Puget Sound versus in the open ocean. And we need a lot of data replication to ensure the accuracy and be able to really compare this data with data from Kunchala Munyasimi and Nunez. In terms of further research, it would be really interesting to look at organisms that are targeted by marine protected area status and what impact microplastics are currently having on them. Iwalai et al. 2024 was a study that I referenced earlier in terms of amphipods and what impact microplastic concentration has been having on them. So it's really interesting to look at that from a biological perspective. Also, again, looking at different marine protected area samples, so collecting from the water column like they did in Nunez versus sediments or bed samples like I did in mine versus beach samples. Also looking at plastic policy evaluation, Washington recently passed a thing that it, bans microplastics in cosmetics and things. And so looking at the different policies that are being put forth by the Plastic Free Washington Committee and what impact that is having on their concentrations. And also looking at other metrics of MPA efficacy, like chemical pollutants, as they did in ABESA 2018, and looking at how other pollutants might actually be higher in marine protected areas as well. And then also looking at biodiversity, which you might hear from if you come to next week's symposium. The conclusions that can be drawn from the study are that marine protected areas cannot protect from microplastic concentrations because there was no statistical significance in the microplastic concentration between marine protected areas and reference sites. And then again, 93% of samples were microfibers, so that is an area of microplastic pollution that we really needed to be taking a look at. These are my references. I have a lot of thank yous. I really want to thank my mentor, Julie Masura at the University of Washington Tacoma Microplastics Monitoring Lab. She really guided me through this project and let me work with her during the summer on learning how to do all the different all the different microplastic processing. And she's actually the one that wrote the study to standardize methods for microplastic testing. So that's pretty cool. Thank you so much to Mr. DeGroat and Mr. Nielsen for guiding me through this project and really inspiring my curiosity as a scientist. Thank you to the microplastic queens. Those are the girls at UW that let me work with them in the lab to process my microplastics. Thank you so much to my family for being so supportive, to my marine camp family, and especially to the Groat Boat OG. What questions do you have? Yes? There. So the question was like, did I look at how currents might affect the microplastic concentration? I did not look at any like specific currents in between the different areas. That actually is a really interesting take, and now I want to go look that up. Yeah. Yeah, there were some currents, and the big thing is also like the tidal exchange, all the different waves like going in and out. That has a big impact on microplastic concentration. Yes, that's a great point. So Mr. DeGroat and some of his friends helped, and my dad as well, helped with the deployment of the Van Veen while we were collecting samples. I did all of the processing of the samples by myself, and then my mentor, Julie, taught me how to do all of that, and also helped me with the statistical analysis and the development of the ArcGIS maps that I used in this. Yes, Mr. Nielsen? Yeah, definitely. Okay, so the first question that I'll tackle on that was, sorry, do you want to repeat the first question again? Mm-hmm. Oh, right, yeah. Mm-hmm. Oh, yes. So the question was like, how would enforcement impact microplastic concentration in marine protected areas? And so the enforcement that was talked about in Christie et al. 2014 is the enforcement of current marine protected area status regulations, which is just the prohibition of the taking out of marine resources. A lot of times that's going to deal with fish and other species. So that necessarily wouldn't have an impact on microplastic concentration, but a lot of times the human activity in different marine protected areas that goes into harvesting marine resources would have an impact on microplastic concentration. And what I'm advocating for through kind of this study is the fact that marine protected area status marine protected status by itself is not enough to impact microplastic concentration. That's what I really wanted to emphasize, that we need to be doing more, expanding the definition of what a marine protected area is. And then your second question was about, sediment. So basically, It's not necessarily a good thing for the microplastics to become trapped in the sediment because that is going to have an impact on benthic organisms. And then as well, sediment is a good place to look at microplastics as a whole because in the water column, a lot of times with tides and currents, you're going to see different concentrations and see a lot more variability in that than you would when looking at microplastics in the sediment. So the sediment can be a good resource to look at what the greater microplastic concentration is in the area. Any other questions? Thank you.
I'll let you go ahead and introduce her later. We'll just do. Hey, we're gonna take about a 7-minute break. That's just time for you to do maybe 15 push-ups, a few burpees, do something to get the blood flowing in the right spot. There is some cookies over there, and you're certainly welcome to them if you haven't already gotten into them. I think you'll need to take the plastic off. So in about 6 more minutes, I'll call you back.
All right. Hello, my name is Clarice Folks, and my research is on the effects of organic and inorganic treatments on the water retention characteristics of soil. So, organic food consumption has increased steadily over the years, with an 8% increase from 2011 to 2021. The most common reasons for switching to organic foods are the possible health benefits and to support greater ethicality in the food industry, mainly pertaining to the negative environmental impacts of inorganic production. California is leading the country in both organic production and retail sales, with an organic industry worth over $14 billion and farmland spanning over 2 million acres. Organic agriculture has been shown to be more profitable than conventional or inorganic agriculture, but it can be further optimized by removing unnecessary or ineffective soil treatments. Decreased startup costs can be an effective incentive for producers to invest the time and money it takes to switch to organic farming. Organic agriculture is not only more profitable, but it is seen to be more effective in regulating soil water retention. Droughts have and will continue to threaten the California agricultural industry and are expected to increase in frequency and intensity over time as a continual effect of climate change. Maximizing water retention in soils is a crucial step in reducing the effects of drought on crop yield. Factors such as soil porosity, or the space between soil particles, play a critical role in increasing soil water retention, which is in turn minimizes water loss due to evaporation. Maximizing soil water retention by optimizing soil structure is necessary to both reduce water usage and maintain a favorable crop output. Because of the increasing prevalence of drought, there's a growing need for higher water retention in California soils. One factor responsible for increasing water retention is soil organic matter, which increases aggregate stability. Soil aggregates are particles made up of minerals and organic matter. The more stable a soil aggregates are and the bigger they are, the better a soil is at retaining water. You can see this in this image, that the aggregates are somewhat broken apart and have filled the spaces where air and water would otherwise be able to move through. In this image, you can see that there's no soil filling the pore space, which allows for water to enter the soil. Soil forms in larger clumps like this when there's more organic matter incorporated within it. In this case, more water can enter the soil, as opposed to a soil with less aggregation, which is prone to compaction and lower water retention as a result. So a soil water characteristic curve, like the one shown here, is a visual representation of the changing water content of the soil as the matrix potential increases. You can see the matrix potential increase as the volumetric water content decreases. This demonstrates how plants have to use more energy to absorb water as the soil dries. There are 3 key values on the graph that I'll be referencing later on. These are highlighted from left to right: the saturation point, field capacity, and the wilting point. These values tell us how well a soil absorbs, retains, and drains water over time. Comparing these values of soil treated with organic and inorganic additions shows us how these treatments affect water retention. So the use of organic matter in soil as a fertilizer is a key aspect of organic farming management. The carbon in organic fertilizers acts as a binding agent, similar to glue, to promote soil aggregation, which ultimately contributes to its ability to preserve and strengthen soil structure. Strengthening soil aggregates counteracts compaction, which is often caused by the use of heavy machinery common in commercial crop production. Studies show that an increase in compaction directly contributes to decreased crop production, so incorporating soil treatments that strengthen aggregates that can help maintain and improve overall crop output. Overcompaction not only decreases crop yield, but also limits root growth and increases topsoil erosion. These detrimental effects make it all the more important that the proper levels of compaction is maintained. When soil particles are more densely packed, less water can infiltrate. And because soil aggregation increases porosity, it's expected that compaction can be mitigated by incorporating organic matter into soil. So the samples used in this study are from a long-term experiment. Such experiments are particularly effective in creating more accurate representations of the changes that occur than when soil systems are implemented outside of the experimental realm, as they significantly reduce the chances of interpreting short-term changes in soil as lasting effects of long-term implementations. Additionally, long-term experiments have brought about new methods of analysis for data sets that have been used in other applications. The most obvious benefit of long-term experiments is that more accurate predictions can be made about future changes in soil with the data collected, as opposed to data from short-term experiments. This would be beneficial in preparing for and adapting to changes before they occur, potentially reducing or eliminating adverse effects and saving money in the long run. The problem that we look to address is that there's limited information on the long-term effects of organic systems on soil hydraulic properties. The objective is to interpret and compare the water retention values of organic and inorganic soils using soil water retention curves. My hypothesis was that soils with a cover crop rotation and organic manure fertilizer will have a higher overall organic content than soils with inorganic mineral fertilizer and no cover crop. The soil samples we used were collected from Russell Ranch, which is a UC Davis research site. My mentor retrieved 9 samples from organic and inorganic plots at random to use in this experiment. These samples were taken to the Washington State University Research and Extension Center in Mount Vernon for testing and analysis. The organic soils were given an organic manure fertilizer and a legume cover crop in the winter, and the inorganic soils were given synthetic mineral fertilizer and pesticides with no cover crop. The soils were collected in 250 milliliter metal cylinders and saturated upside down with the calcium chloride solution for around 24 to 36 hours, until the top of the sample was visibly shiny. Once they were saturated, each sample was placed onto a high prop sensor unit where the average matrix potential of the soil was measured. After around 10 days, the soil was removed from the sensor unit, separated laterally into fourths, and ground into a fine powder. These samples were weighed and put into the WP4C dew point potentiometer. This machine was used to get more accurate measurements of matrix potential than the high prop at lower water content levels. Once we processed the samples, we combined the data points and found the average water content values for each tension point of each of the treatments and graphed it on Excel. The graph on the bottom right is just an example of how the WP4C data points help complete the entire water characteristic curve to get a more accurate representation. Once the graphs were completed, I identified the saturation point, field capacity, and wilting point on each graph. The average saturation point for the organic samples was 55.65%, the field capacity was 34.97%, and the wilting point is 18.16%. The average plant available water capacity for these samples was 16.81%. For the conventional samples, the average saturation point was 51.63%, the field capacity was 33.97%, and the wilting point was 16.33%. The average available water capacity for these samples was 16.33%. So these are the data points for each treatment. The orange one is the organic matter, and the blue is the conventional. I'm not sure if you can see the data points, but you can see how the difference in water content becomes less and less pronounced as the tension increases. Despite this, the critical data points for the organic treatments were still consistently higher than those of the conventional. We can deduce from this that organic additions to soil do increase the overall water content. The available water capacity for organic samples are also higher than that of the conventional samples, which shows that the crops in the organically treated soils had more water available for uptake. This supports past research in that the effects of soil organic carbon on water retention may be less pronounced than previously thought. Despite this, incorporating carbon into soil still has its benefits, primarily being that it still improves soil structure, which ultimately supports plant growth and output. This research shows that cover cropping and organic fertilizer soil additions do improve available water capacity compared to not cover cropping and using a mineral fertilizer. The limitations of this study were that we were only able to process 18 samples in total instead of the intended 48 due to the amount of time it took to process each one, and that the soil we sampled was treated for 25 years instead of the intended 100 due to financial constraints. Furthermore, future research quantifying the porosity of soils given each treatment would give us a better understanding of how these treatments affect soil structure. So thank you first and foremost to my parents for always supporting me. Thank you to Gabe LaHue for allowing me to join his team and work on his research. Thank you to Mr. DeGroot and Mr. Nielsen for leading the marine chemistry program. And thank you so much to the marine chemistry class of 2024 for being awesome. All right, these are my references. Any questions? Yeah. So that was so she asked how, I'm sorry, repeating. Mm-hmm. So these two values are the wilting point and the field capacity and the wilting point from left to right. So the field capacity is determined to be at a tension level of about I forget the matrix potential actually, but it's at a certain matrix potential, and that's why they're at the same tension but different water content values, and that's the same for the wilting point. So, yeah, does that answer the question? Or, yeah. Okay. All right. All right, so he asked if I talked about the type of organic matter that was incorporated. The source of the organic matter? Well, the organic matter was, I believe it was pig manure as a fertilizer, and then the cover crop was a legume cover crop. All right. Thank you. Oh. And the next up is the man of the bees, Christopher Abitis.
Okay. Hello, everybody. My name is Christopher Bitis, and this is my research project comparing formic acid and oregano oil as pesticides against varroa destructors. So, as I'm sure we are all aware, bees play a vital role in our Earth's ecosystems and modern agriculture as the lead pollinators. If they were to die out, plants wouldn't be able to be fertilized and ultimately reproduce, and a lot of the species we now know would be gone. So with that in mind, it was unbelievably worrying when statistics and data showed that these exact bee populations were on a gradual decline for the past couple decades. Some of the leading causes came out to be climate change, poor environmental and agricultural practices, such as using harmful pesticides, but most pressing is the invasive varroa destructor. The varroa mite is a pest that came from the Asian honeybee in the 1980s to America, where it is not native. The mites basically enter the hive by hitching a ride on the bees off a flower, and once in the hive, they reproduce rapidly. It becomes an infestation, and the mites tend to feed off bee larvae and pupae, weaken the bees themselves, and spread diseases across the entire hive, such as deformed wing virus, which ultimately kills the hive off. Now the question is, how do we solve this problem? The most immediate answer is a miticide that kills the mites, yet keeps the bees alive, which in 2013 was oxalic acid and formic acid, two significantly effective organic acids which tended to greatly lower mite infestations within the hive. Now fast forward to 2022, one of those exact same miticides, oxalic acid, is not very effective. The question is, did its counterpart formic acid, which is a similar chemical makeup, follow a decline? Now, formic acid is still used today, and if it has followed the decline, it is important that we stop using it because it is known to be at least slightly toxic to the bees themselves, killing some adult bees, and as seen by this study right here by Oliver in 2022, they have a high chance of actually killing the queens themselves, and if the queen dies, the hive is at a chance to basically be gone. Now, moving away from organic acids, scientists have started to notice that essential oils derived from plants with the molecule thymol in them are highly effective at suffocating the mites and killing them as well. This study right here compared around 30 or so different essential oils in a petri dish, basically put a live varroa mite in there for 2 to 4 hours and drip a couple of drops in it, and a handful of the essential oils such as cinnamon, litsea, and the subject of my presentation, oregano oil, proved to have some significantly high toxicity rates. Zoning in on oregano oil, a study by Dr. Guzman-Novoa in 2017 compared different means of bringing oregano oil into the hives. The focus on this one was a aerated form in which an at-home aerator, like a plug-in for Breeze One, would be filled with oregano oil attached via a extension cord and aerating oregano oil throughout the entire hive. And what this demonstrated was that oregano oil was highly effective in this study at killing the mites, yet keeping the bees alive, with little to no toxicity to the bees themselves. So the gap in knowledge is formic acid still as effective as a miticide, or has it followed similar downward patterns to oxalic acid? And then should oregano oil and other at-home oils be embraced as beekeeper miticides? My initial problem statement was that I wanted to know, does oregano oil outclass formic acid as a miticide when viewed through the lens of mite mortality? So my goal was to compare both oregano oil and formic acid. My initial hypothesis was that formic acid would outperform oregano oil, though oregano oil would still be slightly effective. My second problem statement was I wanted to know if both chemicals had either positive or negative side effects. My goal was to analyze these side effects, and I believe that formic acid, as seen in previous research, would still be slightly toxic, though not to the extent that I would lose my hive. I also hypothesized that oregano oil itself would have no outside influence on the bees. To actually conduct my project, we used Bellarmine's apiary in the back near the greenhouse. We have 2 Langstroth or wooden hives and then 1 plastic or Apme hive. We decided to divvy it up and treat one hive with a constant release of aerated oregano oil, basically a similar structure to Dr. Guzman-Novoa's, except we used 1 dispenser. A wire mesh was placed beneath a super or a category or a box in the actual beehive. The plug-in was ran 24/7 for a multitude of months, and we would keep track of the mites during this time. On the other hand, formic acid in a wooden hive and the plastic hive, we treated when necessary. So if the mite counts were through the roof, we would treat with formic pro sugar patties, basically an amalgamate of sugar, bee wax, and formic acid, of which the bees would walk on and dispense throughout the entire hive, which made my independent variable the treatment method. Now to actually look at the number of mites within a hive at any given point, we used the mite count method, in which you take a poster board or a piece of foam board and you lather it in vegetable shortening before you lay it underneath the hive overnight and take it out the next day. It acts as a sort of glue trap, so any number of mites that fall off will get stuck, and you can count them the day after. Now, it doesn't get all the mites in the hive, but it gives us a good idea of how infested the hive truly is. So we decided to, in phase 1, do a mite check once every 2 weeks from around July 2nd to August 19th, and then once every week from August 27th through October 14th. My initial phase 1 research, only 4 weeks, was used to see when mites first started appearing and if oregano oil, which was still running at this time, would do a good job at preventing the entry of the mites. As seen here, where the number of mites counted in each mite check is on the y-axis, and the x-axis is the date that this was collected. Each hive had mites enter the hive at the same date, the week of July 30th through August 5th, at relatively similar numbers, around 3 to 4. Most continued climbing, mainly the 2-1 hives, however the plastic formic hive dipped after this period. In the second phase of research, in which we monitored the growth of the mite populations, you can see that there is a pretty gradual climb in which oregano oil-treated hive had substantially less mites compared to the other two untreated hives for the most part. However, come to the week of the 24th or the 30th, at which point we decided that around 39 mites was too unsafe to not treat. We decided to treat the 2 formic hives with a Formic Pro sugar patty, which as a result, you can see the sudden decline in the week of October 1st through the 7th. Now, note the indiscrepancy in data on October 1st through the 7th in the wood and oregano line. This was due to an improper mite count. The borders pulled incorrectly, and as a result, it showed less mites than there actually were. However, if you connect the dots, you can still see the positive climb. Now, something important to note, these are the 2 graphs combined, both the appearance and the growth of the mite populations. Mites are pretty seasonal as bees become more dormant and stay in the hive as much, usually in the fall to winter seasons. That is when the mite infestations tend to grow, as there's more available food for them to eat, and the hives are overall more active. Analyzing the side effects, following the formic applications, the bees were definitely less active. This bee does not look very good. It was very lethargic. It was very lazy. You can clearly see the bees buzzing around at any given point. However, when the formic applications were laid out, the bees were not doing that. The release of oregano oil as compared to the other 2 hives, when they were untreated, the bees were not influenced in the slightest. They were so happy. They were buzzing around. It was a very fun time testing on them because it was just great. Now, each hive's bee population did grow to normal rates following last year's project with the bees. Each of the bee populations were pretty low. We even got a new hive this year, so it was cool to see that no matter the oregano oil, each beehive's population of bees did grow at a regular pace. Analyzing the initial phase 1 appearance of the mites, it is clear that oregano oil failed to stall appearance of varroa destructors. It is commonly thought that oregano oil performs better as a preventative method as opposed to a treatment method, but this was not the case. The infestation began at normal rates and proceeded to grow at normal rates, even though oregano oil was being dispensed at this time. The sudden decline of the plastic formic mite count was most likely due to the fact that the mites were first coming in, and because it was late summer, there were fewer of them than there would be in, let's say, winter or fall. Analyzing the actual infestation growth, the oregano oil did effectively lower the growth rates of the varroa infestations, as seen by the fact that the green line is consistently lower until formic acid was applied, at which point formic acid did demonstrate to be substantially more effective than the constant release of oregano oil. Within 2 weeks, it brought the varroa count in the wooden hive from 39 mites to 12 mites overnight, and within the plastic hive from 37 mites to 18 mites overnight, supporting my initial hypothesis that formic acid would still outperform oregano oil, though oregano oil would still be slightly effective. Now, analyzing the outside influences, people might bring up the fact that the hives are different materials, plastic and wooden. We made sure to keep track of the internal humidity and temperature, which is the biggest factor in this material difference, using a broodminder Bluetooth temperature and humidity tracker. We noticed that within all 3 hives, the temperature remained pretty similar between the 3, although the humidity in the Atme hive was slightly higher. As demonstrated by actually my sister's research last year in marine chemistry, this difference is negligible, the material difference, as varroa infestations tend to climb at pretty equivalent rates, no matter the material of the hive. Now, looking at the side effects themselves, the decreased activity of the bees following the formic applications did support my second hypothesis that formic acid was slightly toxic to the bees themselves, although my queen did not die, which I'm very happy about because then it's very expensive. All right, now before I go into my conclusion, I would like to look at my project limitations, namely time, at which point, if we had a bigger time frame, maybe a year or 2, to look at the full seasonal cycle of varroa infestations, we could be able to get a better picture. possibly look into the idea of maybe dosage of oregano oil, the number of dispensers that you have, the application methods. Another significant variable was the number of hives that we had available to us. Each hive is different. If we had 50 hives, we could have gotten a better picture of what the average response varroa mites is to oregano oil or formic acid. Because of this, my project was more so observational as opposed to statistical and mathematical. And then lastly, hive health. The beehives are a big part of Bellarmine's sustainability push, and as a result, we did not want to kill the bees while doing a process or a project where we wanted to save them. So when in a single mite pull, we saw around 72 mites in the oregano hive, we knew we had to immediately treat with formic acid and stop data collection. For context, last year, one of the hives reached around 75 mites in one mite count, at which point that hive, the bees basically just left. Think of it as if your house was so full of rats that you cannot live there. Now, conclusion, comparatively, a single application of formic acid did outperform the long-term release of oregano oil, although due to the negative side effects of formic acid, queen deaths, toxicity, further research is needed to find a better alternative to this chemical. Ideally, one of the essential oils I listed out at the start, or further analysis of oregano oil, is a very great pathway to possibly find a solution. As I mentioned earlier, maybe dosage might play into it. And then maybe the solution is not in oregano oil, but rather in one of the other ones I mentioned, like cinnamon oil or linseed oil. There are a lot of different options. As for my acknowledgments, I would like to thank Mr. DeGroot and the Marine Chemistry Class 2024 for being such great support groups and teachers. I had a lot of fun along the way. I'd like to thank my mom and my dad for being great supporters as well. My sister for introducing me to the bees and introducing me to the whole marine chemistry program. And then my personalized mentor, Mrs. Savage, who was a great support for me throughout the entire time. She taught me the do's and the don'ts with the greenhouse and the bees, and without her, this project wouldn't have happened. And then here are my references. Any questions? Yep. Yes, it is most likely the result of built-up mite resistance, small scale evolution, if you want to put it like that. Same thing kind of happened with the Asian honeybee in response to the varroa mite. Back with the Asian honeybee is the most common honeybee. They are not as affected by varroa mites or the disease that they bring. In the same way, the varroa mites were not impacted by oxalic acid as much over time due to the frequent use, as it was the most common at-home miticide for these periods of time. Yep. I thought about that. We have yet to pull the honey, although I'm looking forward to try it because oregano-flavored honey would be pretty cool and it might sell well. Maybe a future marine chemistry student would like to do a taste test between cinnamon oil-treated hives and oregano-treated hives. That is a possible pathway. Talia? Well, you can buy the queen bees. You can buy the queen bees, the hives. They are pretty expensive. Luckily, Mr. DeGroot and Mrs. Savage did not make me pay for the queens. I would be flat broke if that was the case. I'm not sure the exact number, but I would probably guess it was in the hundreds. Mr. Nielsen? Mm. Well, we did lose a ton of bees over time because of the varroa mites, which is why treating them is such an important thing. As for where the hives get the varroa mites pretty much exist in all of America at this point. They can climb into the hive directly, or most commonly, because they can't fly, they will be found in a flower. And if a bee is coming on to pollinate or collect nectar, the varroa mite will just hitch a ride on its back and then enter the hive that way. The biggest thing is they reproduce within the hive, which is why they become so infested. Maya? The question is, why do we not disperse the formic acid like we would the oregano oil? My answer to that is the sugar patty form is the most adopted method. That is what most people use, and it's what most people have available. So I wanted to make this more accessible if we were to find anything significant. Nobody would really have, liquidized formic acid at their house, but sugar patties are more available on, like, I don't know, Amazon or something. In Dr. Guzman Novoa's original setup, he compared a sugar drip, basically mix some oregano oil with some sugar and water and drip it over the hive, which dispenses it evenly. He also compared the aerated form, which I based mine off of, and then it wasn't as much of a sugar patty, but there's a method where you soak a piece of cardboard in the sugar, water, oregano oil solution, at which point, bees like sugar, they'll walk on over it, similar to the sugar patties, and it will dispense throughout the entire hive. Bryn? Are there multiple species of varroa mites? I'm not entirely sure, although it is my belief that the one that we were looking at is the most common, I believe. That didn't really answer your question, but it was something. Okay, I would like to introduce the next project, Julia Shimshock with machine learning.
Hi everyone, I'm Julia Shimshock, and my marine chemistry project is developing a predictive machine learning model for FRC matches. So firstly, you all have to know what FIRST is. Essentially, it is this global competition in which people of multiple age ranges build robots and have them compete against each other. So I compete at the level called First Robotics Competition, or FRC, which is for high schoolers. And there are 3,300 participating high schools internationally, so it's a pretty huge program. For the different levels of events, there are regionals, districts, district championships, and the world championship. But what's really important to know is that there's qualification matches for all of these, and then like playoff matches. And every team's goal is to get to the world championship, which is pictured here. It's a really huge event. And match assignment starts off as a random thing. So there's 3 robots versus another 3, and this starts off being randomly assigned, but eventually you progress to a phase called alliance selection, in which robots choose which robots they want to play with for the rest of that competition. So how FRC matches are formatted is, first of all, there are 2 alliances, the Blue Alliance and the Red Alliance. And it's in a 3 versus 3 format, so there's 3 robots on the Red Alliance and 3 robots on the Blue Alliance, and they compete to score the most amount of points possible in the 3 different game periods, which are autonomous, teleoperated, and endgame. So the autonomous period is the first 15 seconds of the game, where robots run pre-programmed code, and teleoperated and endgame are remotely controlled by drivers, and there's usually some sort of special task in endgame that is not present in the autonomous or teleop periods. Additionally, the game changes every single year, but these 3 periods are the only consistent aspect of every game. So to create a robot, there are a lot of different things that you have to do. So there's a programming sub-team, a manufacturing sub-team. Ooh, looks like the iPad disconnected. One sec. There we go. Okay. Ooh, let me. Okay, here we go. Okay, so there's different sub-teams it takes to create a robot. So there's programming, manufacturing, electrical, all different aspects in order to have it function. But one of these really crucial sub-teams is scouting. And while they don't work on the robot, they work to advance you in competitions by collecting data of the different robots at these competitions in order to pick which teams they're going to work best with during alliance selection. So they're a really integral part of the team. So there's a lot of different levels of scouting across different FRC teams, because some are kind of low resource and focus more on building their robot, so they'll ask really basic kind of rudimentary questions about your robot, while others have really high resources and are able to track specific data for every robot and can use data visualization tools in order to kind of use this in the alliance selection. So there's sort of this disparity there. And in order to resolve this, StatBotics is a website that was developed that is an EPA model to predict match outcomes. So this expected points added model, or EPA, is essentially a moving average of each robot's performance. So it's how many points a robot is expected to add to its respective alliance. So the prediction is made by summing the EPAs of both alliances, and whoever has the highest EPA will win. This has currently an accuracy rate of 72%, which is pretty good for a machine learning model, but it could improve and therefore be more useful to the teams that need it. And additionally, it doesn't account for a team's history. So if historically a team does not perform well, StatBotics doesn't take that into account and starts everyone off at a level playing field each season. So in order to kind of resolve some of these drawbacks of StatBotics, I created my own program in order to simulate and predict the outcome of matches. So firstly, a problem is that this advanced scouting is limited to elite teams. So in order to make this resource, which is free for everyone, more useful for them, I am aiming to create a model with a higher accuracy rate than StatBotics. Additionally, the current FRC predictive models available, StatBotics doesn't account for historical team data, so my model aims to use data across multiple seasons. And lastly, you can't really strategize based off of this outcome effectively because it doesn't tell you what was important in a team winning. So what my model does is provides an important score for those autonomous, teleoperated, and endgame periods so that you can strategize based off of the result. So, oh, it might disconnect again for a second because it is not loading. Oh, maybe not. I'm gonna try reconnecting to it because it is not loading. Okay, here we go. So how my data is, oh, okay, there we go. So how I'm using this data in order to build this model is I'm starting with a bunch of raw data that I'm pulling from this huge source that I'll mention soon, and I'm moving it into a data cleaning period. So I'm basically filtering out matches and data that aren't useful for the model and are kind of outliers, so it's not going to train it effectively. Then it moves on to this weight finding phase, in which I'm going to build regression trees to output specific statistics for each robot in order to feed it into a simulator that's going to predict the actual outcome of the match. So for the data that I used, I got all my data from a website called the Blue Alliance, which is a huge hub of all this data from the past few seasons of robotics. And I pulled 35,719 matches, and there were 3,564 competing teams across both years. So I used 18,000 of these matches to train the regression trees and almost 12,000 to test the simulator. So in order to prepare my data, I used NIME Analytics program, and first I separated the years, so the 2022 and 2023 seasons, because as I said, each year is different in terms of the game, so they have unique variables that we need to account for accordingly. So my mentor built the model for 2022 and taught me how to do it, so I was able to do it for 2023. So then we move into the data filtration period. It is still not loading on there. There we go. Okay, so then we move into the data filtration period, in which, as I said, we take out the games that are sort of outliers and don't really make sense to include in this because it's just going to lower the accuracy of the model. And then we divided the data into the portions of the game, so the autonomous period, teleop period, and endgame period, in order to assess each of those individually. So then we went into training and developing the regression trees. So first of all, the actual purpose of making these and the outcome that it's going to have is the mean difference. So what this is basically how many points more an alliance is expected to score above the other alliance or below, depending on which alliance the team that we're looking at is on. Secondly, we're looking at the threshold that it's going to take for this team to be predicted to win. So if you have a threshold of 3 points, you need to score above 3 points the other alliance in order to win the match, and if you score below that, you're going to be predicted to more likely lose. Additionally, there's the standard deviation of that mean difference, and also how often that threshold I mentioned is actually met. So the regression trees that I made are predictive of a point difference, as I mentioned before. So it's basically the difference of how many points one alliance is scoring versus the other one. And this was scored from those 18,000 matches that I had mentioned. So then we use k-fold cross-validation in order to validate the model and prevent data leakage. And it's sort of a complicated process, but to explain it simply, when creating these models, all the data is eventually used for validation and testing at some point. And all of this is basically combined to have one model that's really accurate. And in order to make sure that we're not running matches through a model that's already seen them, we make sure to just run it through the fold in which it was used for validation. And then at this point, the weights are generated, so we can move into the simulation phase given those outputs. So again, we're predicting a point difference, which team is going to win, who's going to score more points, and this is where those almost 12,000 matches I mentioned come into play, because those are what we ran through this. And so I used a Python script to simulate this, simulating 200 matches for each of these matches. So it's simulating a prospective match, so the different outcomes of what this could look like. And so to adjust the performance for each robot, because if we just plugged in their means every time to this, we'd get the same result. We started by using the team quartiles. So if a team is ranked very highly and paired with two very low ranked teams, that performance is going to even out accordingly. Additionally, we looked at the variance for their point differences and how often that threshold I previously mentioned was met. So if a robot is very often hitting the point difference necessary in order to be predicted to win, we're going to add the standard deviation more often. But if the threshold is not being met very often, then we're going to more likely subtract that standard deviation in order to accurately reflect a realistic performance. And then we have the output, which is how often an alliance wins in the simulation. So this could be, it's output as a percent. So if a robot wins 56% of the matches in the simulation, they'd be projected to win, and so on and so forth. Additionally, it outputs the importance of each game period, that autonomous, teleoperated, and endgame periods, in order to be able to strategize based off of the outcome. So an example of how this is useful and kind of why it matters is there was one match where our team was projected to win based off of this model, but we actually lost quite dramatically. Our score was 24 points to the Red Alliance, which had 48 points, which for us was not good. It was a very bad match. But after further analysis, we realized that our alliance partner, Stormbots, was not actually at that match, so our performance was much worse. So after realizing this, we simulated the match again with and without the alliance partner. And as you can see, when we simulated with the alliance partner, we were projected to win, but when we removed them, we were way less likely to win in comparison to the Red Alliance. And when this happens, the importance of each game period changes accordingly. So had we been at a competition and we realized our alliance partner isn't going to show up, we could run this simulation and figure out which aspect of the game is going to be the most important in order to prioritize it. So for my results, I'm going to give it a second to load. There we go. For the 2022 weight finders, the autonomous period had a 96.1% accuracy rate, the teleop period had an 89.8% accuracy rate, and endgame had a 97.1% accuracy rate. And for 2023, the autonomous period had a 98.7% accuracy rate, teleop had an 89.7% accuracy rate, and endgame had a 95.1% accuracy rate. And the accuracy rate of the simulator was 76%. So the takeaways from this is that while my model was more accurate, after further statistical analysis, we realized that it is not statistically significant, meaning that the performance can kind of be up to chance. So there's going to be times where StatBotics outperforms us or we outperform StatBotics, and it's sort of variable. However, unlike Statbotics, my model is going to improve with every season that we compete in because more data would mean a higher accuracy as we have more to base off of for all these teams. Additionally, testing in the 2024 season is going to further validate this model for the previous reason I mentioned, but also to test how useful this output of the importance of each period is. And finally, this model can be used by teams to determine the best alliance partners by running through matches and simulating them with potential alliance partners to figure out who they'd like to select during alliance selection. So the limitations of my study, firstly, were that I only had 2 seasons of data that I utilized, which were 2022 and 2023. Secondly, although I do programming for my robotics team, I code mostly in Java, so I have limited experience with Python, so I needed a lot of help from ChatGPT to formulate an effective script. Additionally, as I said, I haven't utilized this in a competition yet, so I don't quite know what that will look like in terms of strategy. So for future research, firstly, I think making an app or website for this is really crucial and making it accessible to the teams that need it. Secondly, trying this with a neural network, which is just a different type of machine learning model, would be very interesting to see if that increases or decreases the accuracy rate. And lastly, factoring in zebra tag data, which is a monitor that your robot receives at the beginning of each competition to track its position on the field. So as you can see here, here's an example of how the robots moved around during the whole thing. And it outputs a timestamp and a position. So you'll be able to tell if a robot dies on the field. And if this happens often, you could use that in simulation by assigning them 0, basically, when they die. Okay, so I'd like to give a thank you to my mentor, Dr. Stephen Shimshock. I knew very little about machine learning before doing this, but he is a data scientist and helped me learn all about machine learning and regression trees and everything, so I'm really grateful for that. Additionally, a thank you to my robotics team because you all have supported me throughout my time at Bellarmine and in marine chemistry, and I really appreciate it. I'd also like to thank Mr. DeGroat and Mr. Nielsen, the program directors, for all the support you've given me, and Mrs. Nielsen, as I really enjoyed the Hawaii trip with all of you. It was amazing. And I'd also like to thank my family for supporting me and listening to my presentation 100 times. And also the Marine Chem class of 2024. I'm really grateful to have spent this time with you guys. And I just have my sources. Okay, are there any questions? Mr. Nielsen. Okay, so the question was kind of how this data will help us select a partner. So the alliances are 3 versus 3, and basically the top 8 teams end up picking alliance partners, and you have to formulate, again, with that group that you've selected, to pick a 3rd partner to be on your alliance. So how this is useful is you could pretty easily and quickly simulate matches with those other teams to figure out who you perform best with or who you score the most points with. No. Yeah, so the question is like, could it be considered like cheating or is it like too effective? I mean, if it got to that point, again, I mean, it would be a resource that we're trying to put out for everyone. So I think if everyone had access to it, it would kind of level the playing field. And additionally, it's sometimes pretty clear who the top teams are anyway. So this would be really useful in determining which of those kind of more mid-ranking teams you'd want to select. All right, thank you.
Let me just double check, make sure we don't have any other presentations tonight. Looks like that's it. if you were here last year, you would have known we've had 2 more. We had 18 students who did 18 different projects, which was great. I mean that. One of the reasons that besides celebrating this accomplishment and the projects with our seniors is for folks coming up to kind of get a sense of what's expected, but also some of the projects that are out there. I really would encourage you, if there's things that are of interest that you see tonight or on Tuesday, come talk to me. We would like to have and continue with a lot of these different things. And you have some experts right now that you can start to work with, and they can kind of tutelage you as well. So, we're going to meet again on Tuesday. We have another variety of projects, and a little bit of a celebration somewhat. I don't know if you know this, but this is Mr. Nielsen's last year in Marine Chem, and also at Bellarmine. At least that's what he says. He's lying about it. He'll be around still. But we're going to celebrate that a little bit next week as well. So make sure you come for that. And I think there are some cookies left. This was courtesy of our cafe. So if you like those, make certain that you go back and grab some. And also thanks to everyone that was helped set things up for today. Our facilities folks, this was live streamed. And so Ben Schmogi, who wasn't able to be here today, made a lot of comments to me about this thing. Ben, if you're listening, thank you very much. And he had a lot of support, you know. So if you need to go back and check out anything or want to go back and review, you can do that too. So be safe on your drive home, and thanks again for being here.